Okay, sorry I'm late. I was having some troubles with one of the scripts. Can you guys hear me?
Yep.
Okay, cool. So our goals today are complex, but I think we can get it all done. Dr. Judd and I have chatted about how lab is going, and basically in the syllabus, the lab has a presentation and a final lab report associated with it. And given how long it's taken to get the data in and get the data rectified between the different groups and things like this, and the fact that we're just now going through the results with you, we feel it would be unfair to ask you to prepare a lab report and a presentation by next week, since next week is the last class we'll meet for lab. So what we've decided to do, you should all have introductions and methods written up for your assignment for today. That was in preparation to get you ready for the remainder of your lab report. And what we're going to do is we're going to still have you exchange papers with one another and read those and get feedback from one another on those to then improve the introduction and methods. What we're going to do is we're going to have you, because you need to be dealing with the results to prepare for talking about this, we would like you to then submit that introduction and methods. And then the results section will just be the relevant graphics that you have for your various questions that you can ask about your data, and detailed captions that are detailed enough that the captions themselves kind of read like a results section. And in the end, what you won't have is you won't have a discussion section, but we'll cover the discussion in the oral presentation next week. And so your oral presentation will still be structured like a normal oral presentation would be structured. It would have an introduction, a methods, a results, and a discussion section. And then the written part you have is largely already written, and it just has the addition of the graphs that you're going to be exposed to today with well-written captions that explain what those graphs mean relevant to what kind of your questions were from the beginning of the semester. That then enables us to assign you a grade for a written report, even though it's not a full report, and it also has a grade for the oral presentation. The nice thing about that is it takes some of the writing pressure off of you over the next week. It seems like a lot of writing going on right now with a literature summary due on Friday, et cetera, et cetera. So in order to take a little bit of the pressure off of you here at the end of the semester, we're going to basically say, okay, we're going to cut it off with kind of the writing that you've done right now, and we're only adding to that kind of writing assignment things that you're already going to have to be processing to get ready for an oral presentation next week. So how does that sound to everyone? Okay, I see a thumbs up. I see a couple of nods. I can't see the rest of you, so I'll have to signal in some other way. Okay, good. Hopefully that'll make the end of the semester seem a little less stressful. So we do want you to have an oral presentation for next week, and we haven't really taught you how to do an oral presentation, and so we are going to go through that today. Let's see. Are you guys on email right now? I have so much running on my computer right now. My computer wants to die. I'm going to send you guys an email really quickly, and I want you to open the file that is attached to it. Okay, there's a email coming to you with a file attached to it. Let me know when you've retrieved it. Just give me a thumbs up on your little response thing there when you've received it and have opened it up. Okay, looks like most of you have gotten it. All right, so this is actually the rubric that we will use to evaluate you when you give oral presentations. And over the course of the 4 years that you're here at Jewel, you'll give a variety of oral presentations in the biology major, and all of them will come with a rubric that basically outlines how you're going to be graded on these things. So for the introduction, you need to have sufficient background information, establish why you're doing this research, think about the previous research that is done that has led to this particular talk that you're giving, and clearly state what your hypotheses are. And then the body of it includes the methods and the results, so the clear description of your methods and how you analyze your data. One of the problems that we often have with students in senior research presentations is they go too deeply into the weeds with the methods sometimes. And so there's this balance between providing the audience enough information without kind of overloading them with unnecessary detail. During this section, it's really important to be clear about what you're talking about. And then the act of describing figures takes some practice as well. And then the conclusions are basically, it synthesizes the results of your study. It goes back to what those original hypotheses were and talks about what your data mean in light of those things. It explains the significance of the study relative to all of that previous research that you talked about in the introduction. And it can also suggest improvements for future study. A lot of times in this class, people get bogged down in the improvements and they don't think very much about the future study part of things. So try not to get too bogged down in the improvement section. In a first year class, there's always tons of things that you could have done better. We'll talk about some of those things today. Some of those things we could have done better because it would have saved me a ton of headaches in analyzing the data. Don't get too hung up on those things. This is your first year. And then there's always a question and answer session. So when you give your presentation the next week, I will ask you questions. Your other members of the class will ask you questions. So are you factually accurate? Do you have a good depth of knowledge about your area of study and things like this? And then there are minor points in this class that are involved for, how is your delivery? Are you enunciating? For your presentation next week, you're going to be presenting as groups. So one of the things that we want to know is, are all of the members of your group contributing equally to the presentation? So figure out how you're going to be dividing up the presentation before you get there. And then how are your visual aids and things like this? Are they attractive? Are they conducive to helping the audience understand? Are they too text heavy? Et cetera, et cetera. And so this is how you will be evaluated on these sort of criteria. And you can see from the weighting of the points kind of how we view the importance of these different aspects of the presentation. So that said, do you have any questions about anything having to do with how one would present? What I'm going to do here in a minute is I'm going to present an actual presentation that I've made at a professional meeting to you and give you the opportunity to critique my presentation. What did I do well? What did I not do well? But before I do that, do you have any questions about the act of presenting?
Since we're presenting it as a group, are we going to be graded as a group or individually about the information we're presenting?
No, you will be graded as a group. Yep. So you'll get a critique sheet, that sheet that I sent you. It will be filled out. Your names are all at the top of it, and your group will get a grade that is based on kind of your overall performance. Yep.
How long is the presentation?
We have guidelines. I think we expect you to be about 8 to 12 minutes or so. Most professional meetings are capped at 12 minutes with 3 minutes for questions and answers. So it's always good to have some time limits. There's a set of guidelines that are in a folder somewhere that I need to dig up and post to you, but you should be shooting for probably no fewer than 8 and no more than 12. And so as you practice with one another, pay attention to time. I always practice presentations before I give them, except for this morning when I did not practice the presentation I'm about to give you at all. And that actually might show through. Other questions before we get started? All right, normally I would do this in a room with a screen with a projector. And I was thinking about actually going up to school and presenting that way. But that's not how you're going to present. So this is going to be my first time presenting something like this over Zoom, but you're going to have to be presenting over Zoom as well. And so as you guys think about your presentations, consider the fact that you're not going to be standing in front of a room. You're actually going to be doing this over Zoom, and you're also going to be handing off the presentation from person to person via Zoom. So having a plan for who speaks when and things like that is going to be important. So can you guys see my screen? Has a lizard on it? Yeah, yes. Okay, good. All right. So normally at a presentation, you would have somebody, can you guys still see my screen?
Yeah.
Okay. Normally you would have somebody present who you are and what you're going to be talking about. So I'm just going to kind of fake that right now. Your next speaker is Paul Kowinski. He'll be speaking about adaptation and acclimation in an introduced species experiencing a northern range extension. Thank you very much. And for whatever reason, I cannot seem to navigate. There we go. So first I would like to acknowledge the spring 2017 herpetology class who gathered a portion of this data. Because I work in cities, I have to recruit the cooperation of police and the Galveston Port Authority Police and the Galveston City Police were kind enough to not arrest me when they stopped me for when I was walking around on the outside of buildings with flashlights at night. My nephew, Ethan Klawinski, came and collected with me sometimes. And my sister and her husband gave me a place to stay while I was in the Galveston area. And I also got funding from the Monte Garmin Fund here at Williamsville College. So normally when we think about temperature relative to organisms, we oftentimes think about global warming. As you can see, over time, temperature has been rising, and especially as a herpetologist working with ectotherms who get their energy and body heat from the environment, we oftentimes think of organisms getting too hot. But one of the other problems with temperature and reptiles is that they can get too cold. So here's a report from the New York Times where there was a cold snap in Florida and iguanas were falling out of the trees because they had been immobilized by getting too cold. Thinking about temperature and novel temperatures that organisms might not be used to encountering is important when you think about introduced species, because when a species is introduced into a new environment, they may leave a nice warm place and go to a nice warm place. So they may leave northern Africa and move to southern Texas. But then as they expand their range, they may expand their range into areas that are no longer warm for them. And in the case of the species that I'm looking at, which is Hemidactylus tursicus, this is one of those species that has moved from warm climates and is expanding its range into colder climates. As you can see here, this is the range of Hemidactylus tursicus. It was introduced in the southern parts of the United States and has migrated and been spread as far north as the Kansas City area, and in this case, Pittsburgh, Pennsylvania, much colder than the native range of this animal in the old world. And so when we think about ectotherms, we think about body temperature influencing performance. And at low body temperatures, performance is low. Think of performance as something like sprint speed. And as you warm the animal up, the performance reaches some optimal temperature range, the performance breadth. And then as you continue to heat the animal up, it finally begins to suffer performance losses because you're basically heating it up so hot that it begins to denature proteins and begins the process of dying from being too hot. Well, organisms can thermoregulate through behavioral means. So here's a lizard in a desert. It can maintain its body temperature at a relatively constant body temperature by moving into the sun when it's cold, moving into the shade when it's too hot, absorbing heat from the sun, absorbing heat from surfaces. So behavioral thermoregulation is important in organisms that don't heat their bodies through metabolic heat production. But I work on an organism that is nocturnal, so it lacks many of those behavioral mechanisms because it's not out during the heat of the day when they can just run into the sunlight when they need to warm their bodies up. And so we can think about seasonal acclimation as one way of dealing with changes in temperature. If you are acclimated to warm temperatures, you might have a performance curve that looks like this. But as you move into winter and you are encountering colder body temperatures, you might shift this performance curve down such that you can perform better at colder temperatures, but what that also means is that you're going to perform worse at warm temperatures. So this is what we mean when we talk about acclimation, when you actually change your physiology to acclimate to whatever the conditions are. Think of mountain climbers going up mountains and getting higher hematocrit, or think about yourself earlier in the fall when we first got those cool temperatures. It got down to 55 degrees and we were all throwing on sweaters. When we get to March and it gets up to 55 degrees, we'll be walking around in shorts and t-shirts because that will feel warm to us in the same way that 55 degrees at the end of the summer felt cool to us. This is another example of acclimation. Another way of dealing with climate is to just have an evolved physiology that handles cold climate better when you encounter cold climate. And so an evolved response would just mean a permanent shifting of this performance curve to cooler temperatures in cold climates, and that performance curve would stay to the right of this figure in warm climates. And so organisms have the ability to both evolve to changing climate, and they also have the ability to acclimate, and teasing this out is one of the purposes of my current research. So on this species, previous work has been done. Huey in 1989 found that the critical thermal minimum, which is the temperature at which if you flip a lizard over on its back, it is unable to turn itself back over, was 10.5 degrees centigrade, which is quite cool. And the critical thermal max on the high end was 41.1 degrees centigrade. They didn't measure preferred body temperatures, but it would have been nice to have that. But they basically found that preferred body temperatures of a variety of other geckos were actually lower than the temperatures that they achieved maximum sprint speed. So that optimum peak was different from the preferred temperatures that the lizards preferred to be at. Hitchcock and McBrayer did measure preferred temperatures, and they found that preferred temperatures of geckos of this species were in the 26 to 28 degree range. And they also found that they only experienced this body temperature in this range during August, so during the warmest time of the year, but they also only measured preferred temperature at a single time. So my questions are about the performance, and in this case, performance of sprint speed of Hemidactylus tursicus, and I want to know if it's changing as it expands its range north in the United States. And I'm interested in whether or not these observed changes are due to acclimation or an evolutionary response, or possibly a combination of both. So I collected geckos in Edmond, Oklahoma, and Galveston, Texas. This is what my study sites look like. My research students tell me I take them to the most scenic urine-soaked alleyways in America. So these animals occur on human habitations. Old brick walls are good for them. And at Edmond, Oklahoma, I collect geckos at the campus of the University of Central Oklahoma, where there is an established population. This is Jacob Marley and Elizabeth Everman, the two people who are co-authors on this presentation with me. So when we get to a wall, we see a gecko, and the first thing that we do is we measure the body temperature of that gecko. And then once we have the body temperature of that gecko, we measure the temperature of the wall right next to that gecko. And then after we've surveyed all the geckos on a particular wall, we take a random sampling of temperatures on the wall, and this is how we quantify the environmental temperatures, basically the thermal environment that is available for thermoregulation for this lizard. Then we bring the lizards back to the lab, and we measure their preferred body temperatures in thermal gradients. There are a bunch of heat lamps at one end of the room. This is a very cold room there in White Science Center. And the geckos basically run up and down these channels. You can see geckos here, here, and here. And they position themselves in places where they presumably are at their preferred temperature. And we come by periodically and we measure their body temperature using the same gun that we use in the field. And then we also race these lizards down racetracks. I've built a laser operated racetrack such that when the lizard runs through these laser beams, they break the beams. That then is processed by this microprocessor, and it sends the data to the computer, and it actually calculates how fast the geckos are running down the racetrack. The racetrack is divided into 4 different segments, and so we have quarter-meter splits for a full meter of racing. So what we do procedurally is we collect the lizards, we bring them in, we acclimate them to whatever the collection temperature is. So if we collect them in the winter, we bring them into the lab and keep the temperatures cool. Then we race them, and then we conduct thermal gradient. We do the thermal gradient to test their preferred temperature. Then we shift them to the summer temperature in this case, because we collected them in the winter. We then shift them to a summer temperature, allow them to acclimate to that temperature, and then we repeat the races and the preferred temperatures again. When we collect them in the summer, we'll bring them in and acclimate them to summer temperatures, and then we cool the room down to winter temperatures. So the data analysis is we looked at sprint speed using an analysis of variance, looking at collection temperature, acclimation temperature, population, and temperature as the predictor variables. And then we also did an analysis of variance on the type of temperatures, looking at population and season to look at what their lives are like out in the field. I could show you all these ANOVA tables and things like this, but I'm just going to show you a bunch of graphs. So one of the first questions you might have is, are Galveston and Oklahoma City different from one another? And here you see the daily highs and the daily lows. The dark red is for Galveston. The lighter red is for Oklahoma City. You see that in the winter, the average highs are lower in Oklahoma City, but you'll also see that throughout the year, the low temperatures, which are the nighttime temperatures, are consistently different between the two groups, between the two populations. So daytime temperatures converge during the summertime, but during the summertime, even nighttime temperatures are different. And that's actually relevant to us because when we look at gecko temperatures, this is the environmental temperature, the random temperatures along the wall. We see that the environmental temperature is very different in winter, but they converge in summer. The wall temperature right next to the gecko is also very different, with the blue being Edmond, Oklahoma, and the red being Galveston. But once again, they converge during the summer. The body temperatures of the geckos in the field are very different in the winter, but once again, they converge in the summer. And the preferred temperature of the geckos actually doesn't really change very much between winter and summer. And so during the winter, the Galveston geckos can thermoregulate within their preferred range, but the Oklahoma City geckos cannot. And both populations are warmer than their preferred temperatures during the summer in both populations. So the thermal environment does present some challenges to these organisms, more so in the winter than in the summer. And so this is, once again, looking at the temperature of the wall right next to the gecko relative to the body temperature of the gecko. And you can see that these animals are what we call thermoconformers. They tend to conform to the environmental temperature. And you can see that the two populations are not really different during the summer. When we look at sprint speeds, what we find is that winter collected lizards that are kept at winter temperatures, so this is winter collected lizards that are winter acclimated. This is the temperature at which we're running races, and this is the sprint speed. And you can see that two populations are different when we collected them in winter and winter acclimated them, with Galveston being consistently faster, even at cool temperatures. But when we warm them up, when we collect them in the winter and then warm them up to summer temperatures, you see that their overall sprint speed goes up. And it goes up similar to the sprint speeds that were reported by Huey. That's what these black dashes are, is the temperatures of Ray Huey's sprint speed races. One of the things that I would like you to note is that we're racing lizards at 10 degrees, which is below Huey's critical thermal minimum value. When we collect geckos in the summer, both populations are performing very well when we summer acclimate them. But when we shift these animals from summer collected to winter temperatures in the lab, one of the things that we see is we begin to see these slight differences between the performance of the geckos at low temperatures. And you'll notice that this performance shifts and switches order when you move to warmer temperatures. So when we take summer collected geckos and shift them to winter temperatures, Oklahoma City geckos do better at colder temperatures, but they do worse at warmer temperatures, while Galveston does worse at cold temperatures and better at warmer temperatures. And this indicates to me that there is some sort of acclimation effect going on, but it's only when you change acclimation from summer temperatures to winter temperatures, because both species respond similarly when you go from winter temperatures to summer temperatures. Just as an aside, one of the things that we also discovered is that the populations are different sizes, with Oklahoma City geckos being actually larger in body size relative to their weight. So they're larger in body size anyway, but they also have better condition in the winter. They weigh more per body length. That's something that I'm interested in looking at in the future. So Hemidactylus tursicus is a thermoconformer. It basically takes on the body temperature of the environment around it. Long-term cold acclimation negatively affects sprint speed, as you saw in the depressed sprint speed values for those animals. Short-term acclimation from cold to warm temperature improves performance in both populations, but short-term acclimation from warm temperatures to cold temperatures has different effects in the two populations. Oklahoma is faster at cold temperature, and the Texas populations are faster at warm temperatures. My speeds are generally slower than those that were reported by Huey et al. And these Oklahoma geckos are larger and in better condition than the Texas geckos, especially in winter. And this is something that a student of mine and I are currently working on, why that might be the case. And that's it for questions. And I'm at 16 minutes, which is like 4 minutes longer than I would be doing this if I was doing it at a conference. But I also didn't practice this before I got up and gave it to you. So do you guys have questions about any of the information in the talk?
Yeah,
As if this were an actual conference. Yeah, go ahead.
About how you figured out the preferred temperature for the geckos.
Oh yeah, so that goes back to the thermal gradients. So they're in a cold room, but then there are hot lights at one end of this. And so if they move down here to the end, they get cold. If they move up here, they get hot. And what you typically do is you find them in the middle here somewhere because they like an intermediate temperature, because this is about 110 degrees Fahrenheit up here, and this is about 50 degrees Fahrenheit down here. And so they hang out in the middle because that's their preferred temperature. So you measure preferred temperature by giving them a range of temperatures that they can choose from, and then you just take their body temperature to see what temperature they've chosen. Does that make sense?
Yeah, that's pretty cool.
And then this is what it looks like when you actually see that. Whoa, went too far. So their preferred temperatures. And you see that their preferred temperatures don't really shift very much from season to season. Other questions?
When you raced them, was there motivation for them to run, or was it just like their nature for them to run down the track?
There is motivation. So we have small paintbrushes. They're paintbrushes like you would use in an artist class. And what you do is you place the gecko in the track here, and after he's in the track, you basically tap him on the tail. And usually one tap on the tail will propel him to the end of the track, especially if it's nice and warm. If it's cold, they may only run a quarter of a meter at a time before you have to tap them again, but you can just tap them gently on the back of the tail. In the Brody papers that you're going to be reading, they prompted the snakes to go down the track by tapping them with a Q-tip swab, a cotton swab. So yeah, you just gently tap them on the tail and they tend to run away from you. So what you want to do is you want to tap them and propel them forward without actually pushing them. And once again, at colder temperatures, at 10 degrees centigrade, sometimes they wouldn't make it down the full meter because they just kind of ran out of steam because their physiology isn't warm enough for them to be performing optimally. Other questions? Okay, what did I do well and what did I not do well in the process of presenting this information?
I got one.
Oh, sorry.
Oh,
No, you go ahead.
Okay, I was just gonna say that I really liked how you had a lot of graphics, like graphs and pictures that we could look at as you were talking and you weren't just reading from your slides, you really knew your information.
yeah, so that's one of the key first things actually is being prepared enough that you know what's coming. And I'm going to show you. Can you guys still see my PowerPoint?
Yes.
Okay, I'm going to go to slide sorter. What's one of the things you notice when you look at the presentation as a whole?
I think every slide has a picture. Almost every slide.
almost every slide is graphical in nature. And so I'm using images to try and get across the information that I'm trying to present. And I'm thinking about how I present it. So for example, this figure here, you know, using color to indicate the things that I'm talking about. Also, the animations that I have, I use animations sparingly, but I use them generally when I want to have the animations help walk me through the progression of how I want to cover something within a particular slide. Other comments? So who made that last comment? Was that Audrey?
Yes, that was me.
Yeah, so another key thing is to be really confident in what it is that you're going to be saying. So, you know, for each of your presentations, for the section that you're responsible for, make sure that you really understand clearly what it is that you're going to be talking about during that section of your presentation. And the other thing is practice enough so that you kind of know what's coming up on the next slide without having to see the next slide. That helps you with flow through a presentation. What else? The good, the bad, the ugly.
You didn't spend very long on each slide, like you moved like quickly, and the next slide correlated with the slide before it.
Okay, so is that a good thing or a bad thing? You're not basically saying whether you like that about the presentation, or did you think that tended to push it along too fast, or?
No, I like that it was fast paced. I like that you didn't like dwell on one slide.
One of the problems of having text-heavy slides is it oftentimes slows you down and you tend to dwell on the text, which is another reason that I tend to do things in pictures. When I have a picture up there, once I'm done talking about that picture, I haven't really left myself anything else on the screen to dwell on, and that kind of keeps me on track and once again, keeps me on time. One of the criticisms you should be having is that I was 4 minutes longer than I should have been, because I've actually given this talk in 12 minutes before, and it took me 16 today. And then you're probably wondering, how the hell did he get that thing down to 12 minutes? But you can. Yeah, I tend to be slide heavy. Back in the old days when I was in graduate school, we actually had to take pictures of slides, and they went in a slide carousel. And I would get to a conference, and I would give them my slide carousel, and they would look at it, and they would have like 35 slides or 40 slides for a 12-minute talk, and they're like, "That's an awful lot of slides." I'm like, "Yeah, it is." But I did this, I had my slides arranged the same way. So whereas an animation that I have here, I click through in one slide, I would maybe have to do that in 4 or 5 slides back in the Ektachrome days. And then they would get finished. I would get finished. I would go back and collect my slides from the slide projector guy, and he'd like, "Okay, now I see why you had so many slides." So, what else? This is your time to criticize your professor without consequence.
On one of the slides, one of your bullet points said something along the lines of, "I am now going to show you my graphs." And I thought
Yes.
that was a little weird.
Oh yeah. So the reason for that is it's the data analysis slide. So I did these analyses, but I'm just going to show you a bunch of graphs because one of the things that people do sometimes is they show you this. This is the ANOVA. This is the first ANOVA that I mentioned in the methods. It looks at how sprint speed varies by collection, by acclimation, by temperature, by population, the collection by acclimation interaction, collection by temperature interaction, acclimation by temperature interaction, collection by population, acclimation by population, temperature by population, a bunch of three-way interactions, a bunch of four-way interactions, and the error term. Now, would you rather see that, or would you rather see that?
The second one.
You would rather see this, wouldn't you? This makes a lot more sense to you, doesn't it? But I talked about this interaction here, and if somebody asks me why I think that interaction is significant, I can go to this line right here, because that's a collection by acclimation by population interaction. And it's not significant in a strict sense, but it's right on the edge of being significant. And so if somebody does ask me about that, I can go to this ANOVA table and show them that. But if I just shot this up on the screen, people would look at me and be like, okay, dude, what are you doing to us? So I basically say here, I did the tests that back up what I say, but I'm not going to go through this because this is way too complicated. I'm just going to show you a bunch of graphs. And these are the graphs that are important to these analyses. So it is a little weird, but yeah, I can see why it would be off-putting if you're not used to listening to talks. But you're probably grateful that I did this and this and this and this rather than this and this.
Yes, it makes much more sense now.
Okay, yeah. And notice that I have these things in my presentation, even though my intention is to never show them to you. They're just there in case I need them. What else? Did you notice how I talked about graphs? When I talked about graphs.
Yeah, I'm a little confused by this graph. I think
Okay.
on the graphs in general, I think you went a little bit quick. It's like there's a lot of information on these slides, and
Yeah.
it's easier to understand than a table, I think, but I'm still a little lost on what some of this stuff is telling me.
So temperature is on the x-axis. The number of individuals is on the y-axis. So these are just frequency histograms. The top frames are environmental temperatures. These are the temperatures that we took all along the wall, the random temperatures. And in the wintertime, those wall temperatures, those random environmental temperatures are colder in Oklahoma City than they are in Galveston, but in summer they converge. The wall temperature is the temperature that we took right next to the lizard after we took its body temperature. And once again, colder in the Oklahoma City population in the winter, warmer in Galveston, but they converge in the summer. Body temperatures are actually the body temperatures of the lizards on the walls, colder in Oklahoma City in the winter, but they converge in the summer. And then the preferred temperatures are the temperatures that we took in the thermal gradients, and those don't vary by season, and they don't vary by population. So what that shows is that geckos, especially in Oklahoma City, are experiencing temperatures far outside their preferred range during the winter, and they're also experiencing temperatures outside their preferred range in the summer, but both populations are experiencing temperatures outside their preferred range in the summer. So yeah, I guess I could have walked through that a little more carefully, but just now, how did I walk through that? What did I start doing? What did I begin with?
Just reading the labels on the axes and stuff.
Yeah, start off by saying, what's the x-axis, what's the y-axis? Then what do? I start then talking about the data. Same thing here. Temperature was on the x-axis, speed is on the y-axis, and then these are just winter collected versus winter acclimated, winter collected versus summer acclimated, summer collected, summer acclimated, summer collected, winter acclimated. And the red and the blue are the two different populations. So when you're talking through a graph, it's always good to begin with the axes. Then if you have a segmented graph like this, begin with the divisions and then start talking about the data. What's another thing that you notice about all these figures? How do I use color?
Is it consistent through your whole presentation? Like you separate the location based on color?
It is consistent. Texas is always red. Oklahoma City is always blue. Why did I code those red and blue?
So we could follow like the same color throughout 3 different slides and 3 different graphs.
Yeah, so it keeps me straight, first of all. It keeps the audience straight. And I chose red and blue because we oftentimes associate red with heat and blue with cold. So once again, this purposeful way in which I've thought about how I'm going to present the information to the listener. So can you just see the full screen or can you see the presenter view?
We just see the full screen.
You can just see the full screen. So I'm going to go to presenter view. It means I have to share screen in a different way. All right. Okay. Let me see if I can do this. When you present in Zoom, you're going to have the opportunity to see things in presenter view or not. And so that's actually not what I want to do. All right, I want to do this. So can you guys see prairie dogs now?
No, I think we're still on the other slideshow.
Oh, really? Can you see prairie dogs now?
Yes.
Okay, so hopefully this will show you the presenter view. Can you see presenter view now?
Yep.
Okay, so this was a slide. This was a slide that a research student of mine borrowed. And when he borrowed it, he wrote a bunch of notes in here. And oftentimes students will do that because they feel that if they have notes, that it's going to help them. But once again, if you are presenting, if you're showing me the full screen to the Zoom audience, but you're watching the presenter view, you're going to tend to want to read this entire paragraph of notes. And what you're going to do is you're going to find yourself getting bogged down in the same way that you would have gotten bogged down if you had put all of this text on a single slide. And so I realize why students put notes in their slide presentations, but oftentimes it ends up trapping them. They get trapped by the text that are in the notes, whereas what they could do is they could just pay attention to the animation that kind of walks them through the information. Start out in Africa, we come to southern Texas. Oh, it's nice and sunny in Africa, it's nice and sunny in South Texas. And then things start migrating around, spreading geographically. And when you spread north, you go from nice warm places to colder places. All that same information is over here, but it's also embedded in the slide itself. And what we'll oftentimes see students do when they do this, when they're looking at the presenter view while they're projecting a slide on a screen in a room, is that they'll get bogged down in the text of the notes and they'll forget to click the button to move the animation along. So they will have read all the information and then they'll go to advance it to the next slide. And instead of advancing to the next slide, it doesn't advance to the next slide, it advances to the first animation on that slide. And then they have to click through that, but they still feel stupid clicking through that without saying anything. So then they repeat the things that they've already said, and it just kind of devolves into chaos. So you can use notes if you want, but I would suggest that you use them sparingly and try not to get bogged down in them as you're there. What else? Any other comments about this?
I kind of liked how the slide went on how you progressed it to whenever you made the flow chart from how you took the temperature, acclimated, and then raced, and then you took another temperature, acclimated, raced, and the whole slide went the same way too as well.
Yeah, so once again, I'm trying to think of how I can organize information for the listener. Presentations don't kind of fall out of thin air. They don't get cobbled together, they get designed. And so the act of designing a slide presentation is trying to put that presentation together in a way that gets the information across to the audience in the most logical manner and in the most efficient manner, if that makes sense. Any other comments? Has it been helpful to see a presentation and kind of pick it apart? Okay,
Yeah,
I've got a thumbs up from Michaela. Yeah, Keaton?
I think it helps to like know what you would expect on a presentation.
Okay. Anything else? All right. So this is what you're going to do for the next week is you're going to work on a presentation that you'll present in class, in lab on Tuesday of the next week. And it should have an introduction that orients your listener to the project that you're working on, the questions that you have, alerts them to relevant literature. So I've now taken that down, but let me put it back up. Notice that in my background information, this is one of the few places where I had text, but it's in the service of basically communicating things that are in prior studies. Now, I would have preferred to actually present this not in text, but in figures, but many of the things that I'm referencing on these two slides, they didn't have figures in these two papers that illustrate this. And so I'm left with text. But in the event that I had a figure of data similar to the figures that I showed you, I would have actually preferred to use those to communicate the information in this prior work stuff. Notice also that I referred to Huey's paper there, and then you'll also notice that I pointed out, although it was fleeting, I pointed out that on these graphs, the sprint speeds of Huey's races with this species back in 1989 are these dark dashes. And I mentioned in the conclusion that generally speaking, my sprint speeds were slower than Huey's sprint speeds, but Huey was working on a group of geckos from Big Bend National Park in the middle of summer. And so it might be expected, just because of their acclimation, that they might have been running faster, because Big Bend is really hot. It's a desert. So I've even done little things like putting prior data from other studies on a graph, even though the paper itself didn't have the graph. I just pulled the data out of tables and put it in a graph. All right. Now, speaking of data, we have prepared scripts. So Dr. Judd and I have been working furiously to try and get the Tuesday and Thursday classes data sets together, and getting scripts written after that's done. Some of the scripts we couldn't work on because we didn't have data. And then the data that we did get seemed messed up in a variety of ways. So, but we've now got scripts written, and I think we've gotten the data about as good as it's going to get. So what I want to do is I want to take each group through the analysis for their statistical analysis, but I want all of you to hang out for all of it. And the reason that I want you to hang out for all of it is because you may see things in other people's data that you might want to comment on and include in your presentation if you find that it's relevant. And so I want you to kind of all hang with us through all of it. You can relax a little bit when we're talking about another group's data, but then you need to be very on task when we're talking about your group's data. And so these data haven't been posted yet because I was actually still working on one of the scripts, which is why I was a couple of minutes late. And actually, there's still a little problem with that script, but it's the very, very last thing of that script that I can't figure out why it's not working, because it was working at 7:15, and then the result was weird and it didn't make any sense. And it's because one group reported inflorescence mass in milligrams and another group reported it in grams. And when they were in different units, it gave us an analysis and a figure, but it was wonky. And then when I went back and converted them all to milligrams, it didn't do the analysis correctly. So I need to figure out what I screwed up when I went and converted units. And it seems like converting units shouldn't be that big of a deal. But what we'll do is I'll run you through all of the analyses, pay special attention to yours, and then shortly after we're done with lab, I will post all of the data and the scripts to the Moodle site, and you'll be able to find them there. So Dropbox is how Dr. Judd and I communicate with one another, it seems, nowadays. So what we did originally, can you guys see my screen with my file structure up? Yeah, okay, good. So what we did was we divided things out, and so each group has its own folder. But then we had some groups that had Tuesday and Thursday lab information. And so what we did was, over the last couple of weeks, we've been basically combining this data into combined data sets that combine all the data from both classes. And so I'm going to begin with pollen load, because pollen load is the simplest data set and the simplest analysis. And also the pollen load group, we've already been through their analysis. And so that's going to be relatively straightforward and quick. So in this folder here are all data and all scripts. And so I'm just going to pull up the pollen load analysis script. And hopefully it still works. I guess we'll see. All right. R is loading. Can you guys see R? I can't see you. Okay. So this is your standard R workspace. The actual script is in the upper left-hand corner. The code that you're executing is down here. This is your global environment. It shows you kind of what your history is. So you can look at the history of all your work. And I've been busy in the last 24 hours. And then over here is where you have all of your packages and which ones are activated and not, and it also shows you any plots that you've generated. So we're going to be looking at this pane down here a lot. So the first thing you want to do is you want to make sure that you're in the right place. That's what Get Working Directory does. And so you want to make sure that you're where you think you are. So I am in my Dropbox account in the Biology 133 to 2020 folder, in the lab data folder, in the all data, all scripts folder. And that's where all my data and all my scripts are. So if I reference anything from these folders, it's going to find it. If you aren't where you want to be, you can use set WD to set that that path to where your stuff is. But I find it easiest just to open the script up in the same place where my data are. So my advice is, when you download these things off of Moodle, put them in the same folder with one another. Put the script in there, put the data in there, and then you'll have it all there together. So this line here reads in the data file. So the data file here is pollenload.csv, and I'm just going to shorthand that DAT to make typing easier. I always look at the data to make sure it is giving me what I think it is. In this case, we have different transects, we have different plants, we have species, we have insect number, we have the raw count of the pollen that they looked at, the patch area of the patch that they collected this from, and the pollen load. And if I want to, I can see the whole data set, and it's got 34 different insects, honeybees, bumblebees, goldenrod beetles, one fly, and a small number of paper wasps. The next thing you need to have is you need to have various libraries that you're going to need to look at this. And if you haven't loaded those already, then you can load packages. You can install packages over here in the packages frame. But I think you probably should all have these available to you. The first thing that I want to do is I want to check that transect and plants are being treated as factors, but they're actually being treated as integers. So we can actually just change them to factors and check them again. And now they're being treated as factors. So once again, I'm executing things up here, and the actual result is down here in the lower left. The first thing we want to do is there's one dipteran that they collected, and you can't really do statistics on a sample size of one. So the first thing that we're going to do is we're going to drop diptera. So you can see here that there is a single, so I just did summary dat. Summary dat basically just summarizes each of your variables in your data set. So there are 12 observations from transect 1, transect 2 has 12 observations, transect 3 has 1, transect 4 has 7, transect 5 has 12. There's six honeybees, 5 bumblebees, 18 goldenrod beetles, 1 dipteran, and 1 vespid. And so this next line just gets rid of that 1 dipteran because it's just going to mess things up as we analyze things because there's only one of them. So when we do that again, you can see that the dipteran is no longer in the data set down here. So we've gotten rid of it. And so the first thing that we might want to do is just do a basic box plot to get a feel for, what our data look like. And what we see is that there's a severe outlier in the bumblebees. There's one bumblebee that had just a ton of pollen on it. Well, it's nice to know that from the get-go, because you know that that's going to be problematic before it's all said and done. So this group is just going to start off with an analysis of variance, looking at how pollen load, the amount of pollen that they carry, varies by species. And so we do that, and we see that species doesn't have any effect. And part of that is that this bumblebee thing here is really variable, and so it obscures all of the other differences that you might see. And when we do a residual plot, we'll see once again that all the other data are fairly normally distributed. It's just this one data point out there that's really wonky. And so we can also test homogeneity variances, and we'll find that there's not significance. But when we do a plot of this, we once again see kind of what we expect, which is that the bumblebee is really variable compared to all the rest of these because of that one bumblebee that has a lot of pollen on it. So once again, we're going to drop the outlier. And so we're going to basically just drop any insect out of the thing that has a pollen load of greater than 20,000 pollen grains. And once again, we see that now the pollen load has a maximum value of 9,000, whereas before it had a pollen load of 24,000. So we effectively got rid of that outlier. We can now run the analysis again. And now we see that there are differences, mainly because we got rid of all of that variability in bombus. Tukey's on a significant difference basically tells us which ones are different from which other ones. And actually, it turns out that the only ones that are different are the paper wasps are different from the goldenrod beetles. But once again, there's a problem here because you have really different variances, very little variation in the goldenrod beetle and a lot of variation in the vespid beetle, but it's still registering that difference as significant. None of the other things are significantly different. We can look at the residual plot once again, and we can see that the data are considerably more normal than they were before because we don't have that big outlier. And when we make a plot of it, yeah, this is what the plot looks like. And so this is not different from that. This is not different from that. These three are not different from one another, but this one is different from that one based on the Tukey's on a significant difference test. Now, one of the other questions that they had was they thought that the area that the area of the patch, how big the patch is, might affect pollen load. And so this is a regression looking at that. And you can see from the p-value here that that regression is not significant. If you want to be convinced that it's not significant, you can make a graph of it, and you can see that it is not significant. And it's largely not significant because they have two different types of patches, and they're both relatively small patches, and these patches vary a lot in terms of the number of pollen grains on the various insects are. And so patch area isn't really having much of an effect, and insects really aren't having much of an effect. Most of the insects carry relatively small pollen loads, and it could be that these pollen loads are low because we were collecting these insects late in the year. It could be that they just don't normally carry a lot of pollen even earlier in the year. Hard to tell because we don't have data from earlier in the year. So that is the pollen load analysis. Questions from the pollen load group about that? Once again, we've already been through this with you, so this should be familiar. So remember that when you're done, if you want a graph, you can save that graph as a PDF. It will save it as a particular thing. So we could save it as pollen patch or something, just give it a name. You can tell it what dimensions you want it to be in, and then it will just save that as a PDF. So and you can ask it to view the plot after you're done. And when you get it, it will look like a figure. And then you can easily import this figure into your Word file. And this is the figure that you would have with a well-crafted caption that describes what this figure is telling you about whatever questions this figure has to do with. And sometimes after you get the figure in its final form, you discover that there are things about it that you don't like, and then you go back and tweak the code and then generate a new figure and overwrite this figure. But those are relatively easy things to do, because as you can see, you know, in R, depending on how we have our windows arranged, that changes the appearance of the figure. And so it's not going to necessarily look the same when printed out as it does in the graphics pane here. So you always want to look at it before you kind of leave it behind. And you can scroll through whatever figures you have generated using these arrows. It just takes you through whatever figure you have. You see that here Bombus overwrites, the species names are overwriting one another, but when you blow this up bigger, when you save it as a PDF, those things won't be overlapped. So, questions? Okay, then let's go and do the pollen limitation analysis. So this is going to be the pollen limitation group. I'm going to clear out all these figures so that we don't get confused. So there's a little broom icon in the corners here, and right here, and you can use that to basically sweep up your workspace. So once again, check your working directory, load whatever packages you need, and then this is the pollen limitation data set that has both the Tuesday data and the Thursday data in it. And you can either get it to do the whole data set, but this data set is bigger, so it's reached the maximum that it will put out. And so actually a better thing to do is just to look at the summary. And so you see that you have lab, patch, plant, stigma number, plant height, the full standing up. So this is plant height as it stands, and this is plant height when you've straightened it out. You've got inflorescence length, patch size, patch density, interpatch distance, the amount of pollen that are from other species, the amount of pollen that are from self, other and self with one added to it, because to calculate this ratio, you can't, why is that there? You can't divide by a 0, so we added 1 to every count in order to be able to do the division. And I'm going to go and fix this data file because there's something in it that I don't like. It's giving me 2 columns that don't have anything in it, and I just don't want to mess with it. So I'm going to go over here. I'm going to delete those columns. And I'm going to resave that file. And now I'm going to go back and I'm going to re-import it. And I'm going to get the summary again. And now those two, there were these 2 weird X and an X.1 with a bunch of no data in them. And now those are gone. So that's what I just did there. Attaching a file just makes it easier to work with because all you have to do is type in the column heads rather than the data frame and the column head to get access to a column. So once again, it's treating some of your numbers as integers, and we actually want to treat them as factors. And so this just changes those things. And then this is a common formatting block for creating graphs, just so that all the graphs look the same, because we're going to be generating a lot of graphs. So one of the first questions that you guys might have had was, does the pollen count differ by patch? So you had these different patches that you collected from, And so you can think of each of those patches as a separate unit, and you can calculate the average number of pollen in each of those areas. And so what we see is that patch actually makes a big difference. So patch is significant, and we can generate the means for all those patches. So you have something is wrong here, because there should be 19 patches of data. I didn't override it. All right, 16, 17. I've changed this, and somebody changed it back. So it's always good to look at sample sizes, because what this basically is telling me when I look at this is that there are 16 patches, but I know that there are actually 19 patches. So I have to go back and reload the data file. When I look at patch, there should be 19 patches. Thank you very much. So now we can go back and do this. That's the reason it was giving us more data. So there aren't differences among patches, as it turns out. And what we see is, when we look at this, is that one of the reasons that there aren't differences among patches is, once again, the patches are very variable. Some patches have low numbers with hardly any pollen in them. Some patches have more pollen, but one of the consequences of having more pollen is that the patch is within itself more variable. So we can look at the ratio of pollen from other plants versus pollen from yourself. That's a measure of how much pollen you're getting in from other areas. We see that doesn't differ by patch. And once again, we can plot what that looks like. And it doesn't differ among patches, once again, because that ratio is variable in the same way that the original data are variable. So there's just a lot of variability. So then you might ask, well, do patch characteristics have anything to do with the amount of pollen that you have? And so you can look at patch size and how that influences the pollen count. And in this case, the patch size is a continuous variable, not a categorical variable. And so you would use a regression for this. And so this calculates a regression model. This is the slope value, so there's hardly any slope to this. And as a result of that, it's not significant. And so I've just recorded that as no relationship. But you could make a plot of this. So there are plots down here that you, if you want to, you could copy this graph block here, and you could move it to up here. And when you move it to up here, you can just paste it in. But in this case, you're not going to use patch density, you're going to use patch size. And the Y variable is still the same. So now you need to change this to patch size and this to patch size. And when you do this, it should give you the same graph that you would have gotten down below, except that this is for patch size rather than patch density. And so you see that everything's just really super variable, and there really isn't any relationship between the size of the patch that you're in and how much non-self pollen you're picking up. Except that so then you could also do the same thing with the ratio. So this is just looking at the ratio. And once again, you see that there's no relationship between patch size and the ratio of self to other in the same way that this figure didn't show any differences, and this figure doesn't show any differences. This figure doesn't show any differences, and if you graphed model 4, it would also not show any differences. You can also look at the density of plants per patch. Now, if you go and look at the data set, there are a number of ways in which the Tuesday and Thursday lab groups don't match in terms of the data that are in there. And so that's one of the many caveats that you have to make when you're analyzing these data. And also it appears that one lab tended to get more mature stigmas to look at, and the other lab got more immature stigmas in their data set. And once again, in this case, patch density does seem to have an effect. And so to look at what that effect is, so the p-value here is very low. So we can actually add a little note. And so one of the reasons that there's a graphing block down here is that we want to actually see what the nature of that relationship is. And so there's a slight upward trend such that the more dense the patch is, the more non-self-pollen grains that you get. And so we should see something similar with the ratio, but it's not statistically significant, but it might also be worth plotting the plot. And you see that the plot, the slope of the ratio isn't quite as high as the slope of the actual counts of the pollen grain is. And probably because the ratio is on a much lower scale than the pollen grains are. So this is the ratio scale, and it has less variation because it's a ratio. So you might then ask yourself, well, what other things might be responsible for this variation in the amount of pollen? And so you can look at whether plant height has anything to do with it. It doesn't. You can look at whether that affects the ratio, but you shouldn't expect anything different. You might ask if the distance between patches affects how much pollen is getting to you, because that might influence, the ability of the insect pollinators to make it from one patch to another patch. And once again, you're going to see that there's no relationship on that front. And the same thing would be true for the ratio. You might ask yourself, well, maybe just some flowers have more pollen in general, and they have more pollen that is self-pollen and more pollen that is non-self-pollen. And what you're going to find here is that once again, that doesn't show a relationship either. And you might expect that larger inflorescences might have more pollen that is self rather than other. And so once again, that actually does matter. And so maybe larger inflorescences attract more insects and they bring more pollen as a result of that. But when we look at it, it's actually a negative relationship, such that larger inflorescences actually have less pollen from other species, and so or less pollen from other plants. And so this is what you see. But once again, you see that a lot of this is due to the fact that some of these inflorescences are very small. Or a lot of these inflorescences are very small, and so this is weird. And this is an artifact of the fact that one group measured inflorescence length and the other measured floret length. So you should definitely not include this in your results, because this is a significant statistical result, but it comes from a measurement error that it's impossible for us to go back and correct. So in some cases, people measured the entire inflorescence, and in other situations, people just measured the little florets that they had that their flowers were located on. And so these are all florets, these are all inflorescences, and that's what's leading to this significant result. So this last relationship, even though it's significant, it's meaningless because it's just a measurement difference between the Tuesday group and the Thursday group. So as you interpret these data, keep in mind that these two groups did do things somewhat differently. The Tuesday lab tended to measure more immature flowers, more immature stigmas. the Thursday group tended to search out for the more mature stigmas, and as a result, that will be affecting the pollen counts that you have. Questions about that? That when I was talking to Dr. Judd about this data set in particular, he said that right there is the biggest caveat for the pollen limitation data set, is that there were lots of differences between how the two groups did things. And it was a niggling, detailed sort of task we gave you, so it's not really all that surprising that there were differences. Questions from the pollen limitation group?
So because there was a lot of no relationships based on the data we found, when we're presenting, do you want us to present talking about specifically what we found, or do we also bring in what the Thursday group did?
No, so this is an analysis of the entire data set. So Thursday lab and Tuesday lab are both in the data set. And so this is our best answer with all of the data that we possibly have to bring to the table to answer the questions. And so what you found was that, one, there wasn't a ton of pollen. I mean, the number of pollen grains that you have, just when we go back and let's just go down here at line 172, I'll summarize your data file here. When we look at the amount of pollen varies from 1 pollen grain to 41 pollen grains. In the pollen from other plants, and self-pollen is 1 to 30. So there's just not a ton of pollen on any of the stigmas that anybody looked at. And then when you start looking for differences among patches and the effect of patch size and things like this, once again, you're not seeing those because there's just not a ton of pollen out there being moved around. Now, part of that could be that you were gathering these plants kind of at the end of the flowering season, and so there was probably less pollen in general. So had you done this a month and a half or 2 months before, you might have seen higher pollen counts overall. But one of the conclusions that you might conclude from your data is that these things might be pollen limited simply because none of the stigmas had a ton of pollen on them. And that's borne out by all of your data. And whatever strategies goldenrod might have for attracting pollen from other goldenrods to itself, those strategies aren't really working. If one of the strategies is to grow densely in a group so that you have more individuals right there around you to exchange pollen with, that's not really having an effect. If it's having patches that are closer together so that insects can move easily from one patch to another patch, bringing pollen with them, the interpatch distance didn't have an effect. If the distance from an edge, if things are being wind pollinated perhaps, which they're not in this, but if being next to an edge makes you more visible, or if being taller makes you more visible, all of these things might increase the pollen that you would have available to you, but none of those things had an effect either. So it appears that these things are, at least at this time of year, are pollen limited because there's just not very much pollen on any individual stigma. And none of the things that you might think of as a aspiring botanist that might, in your head, improve the chances of being pollinated, none of those things seem to be having much of an effect. Does that make sense?
Yes, I understand now.
And that's fine that you didn't find effects. It just makes you have to go back and kind of reconsider, okay, if a goldenrod's goal is to get pollinated and it's not doing a very good job of getting pollinated, then as you'll see in a minute, then how the hell are these goldenrods setting so many seeds? Well, one of the things that you're going to find in the life history data set is that only about 80% of the seeds that the plant actually produces are viable, which means that the plant produced the seeds, but the seeds aren't actually, they don't possess a fertilized embryo in it. And so there's a certain degree to which they might be pollen limited, which might account for the fact that only 80% on average of the seeds that are produced are viable. And this is why it's important for you to be thinking about other groups' data while you think about your own data.
Okay,
Other
thank
questions?
you.
Okay, yeah, other questions? All right, life history group. So let me clean up my space. Life history group, you should just already know that your analysis is going to be a long slog, because the nature of life history data within a species is you're looking at correlations between variables that you think might be important. So when you think about reproduction requires energy. Energy can come from a variety of sources. Energy can come from sunlight, and so that's one of the reasons that you guys measured leaf area. Energy can come from stored energy that is in the roots from year to year, because it's a perennial plant. And so if a plant has stored a lot of energy in its roots, it might have more energy to put both into growth and reproduction in the following year. And so what we're going to be interested in is how does things like leaf area and the overall size of the plant influence things like seed set and total seed production and stuff like that. And so that's what this analysis is hopefully going to do. So the first thing you do is load up your libraries, and then you get your data set. And in this case, the Tuesday lab did a much better job than the Thursday lab of gathering data. So most of the data that are in the data file are actually from the Tuesday lab. So we're going to attach this. Now, some of the things that are in your original data file aren't as useful to us. So for example, you reported the mass per 100 seeds, which is what we asked you to do. So you measured out 100 seeds and you weighed that, and you reported that aggregate mass, but we actually want to know what seed size is. And so we can actually just within R, we can get seed size by dividing the mass of the 100 batch of 100 seeds, divide that by 100, and that will tell us the actual average mass of each individual seed. So we'll do that. You measured viable seeds and non-viable seeds, so we can get the percent viable by just dividing viable seeds by the total number of seeds. And then the seed set, we would calculate by basically counting, multiplying the percent viable times the total amount of seeds that you estimated when you measured those seeds by volume. And so this is just, I'm using R to generate some other variables that we would find useful. And so now you see that we've added a seed size, percent viable, and a seed set variable. And you see that the seed set variable here is lower than the total estimated seeds that you calculated when you measured seeds by volume, because this is only 83% of the total seeds based on the percent viable that you have. Similar thing here, only 78% of seeds from this plant were viable, 89% of seeds from this plant were viable. So there's a certain percentage of seeds in every plant that are not viable because they didn't get fertilized. So then we just start down the path of thinking through how aspects of the plant might influence reproduction. So the first thing that you might think is that taller plants would also be bigger plants. And so we can look at the height of the plant and see if that is correlated with the mass of the plant. And one of the things that you see is, of course, that's true. Here's your common formatting block, because we're about to make a plot that shows you the height of the plant and the mass of the plant. And you see that there's a lot of variation in height, there's less variation in mass, but that's a relatively good relationship. There's one plant that has really high mass relative to its height. So it has a really high positive residual. And so mass might be a better thing to look at. So we might look at, for example, plant height. Maybe there's something that has to do with the linear length of a plant. There's a gene that controls lengthening. And so plant height might be correlated with inflorescence length. And if inflorescence lengths make them have more flowers, then you might have more flowers and as a result have more seed mass. And so these are the kind of causal relationships that we're exploring. And that is significant also. So taller plants also have longer inflorescences. Turns out these are not significant. Well, that's not right. They are significantly correlated. This is an older analysis before we combined the two data sets. So I'm putting notes in here for you as we go. All right. Yeah, so taller plants have longer inflorescences. But also you might expect bigger plants to also have longer inflorescences, which they do. Once again, the p-value here is showing you that. But notice also that the R-square values, none of these R-square values are particularly high, 0.31 or 0.32. Plant height is only explaining 32% of the variation in the, total dry mass is only explaining 32% of the variation in inflorescence length. So that's why there's so much variation around the line, is because it does describe inflorescence length, but it's not necessarily describing all of the variation in inflorescence length. And so since we were thinking that mass might actually be the most useful thing, a lot of what you're going to see from here on out is just mass. So does the total dry mass of the plant, is that correlated with the dry mass of the inflorescence? And that's super significant. And that actually explains a lot of the variation in the data. So the dry mass of the plant explains a lot of the variation in the dry mass of the inflorescence. And this is what that looks like. And so that R-square value is higher because these data are more clustered around the line. And this data point here is that data point that was right here, okay? So if you have bigger plants in terms of mass producing bigger inflorescences in terms of mass, then maybe those inflorescences are capable of producing more seeds because they're simply bigger. And so we might want to know if the dry mass of the inflorescence is actually influencing the total seed count. And as it turns out, the size of the inflorescence does not influence the seed count. So larger inflorescences don't produce more seeds. And if you doubt that that is the result, you can just look at the graph of the relationship, and it looks like there's a relationship, but that relationship is largely being driven by this one point that is, once again, a very large inflorescence for whatever reason. So you'll also see that there are fewer points here because this indicates a situation where the Tuesday lab and the Thursday lab didn't have data for these variables for everybody. And so you're going to see that this has fewer data points than, for example, this did, because the combined data set isn't complete. So do larger inflorescences produce higher seed set? And in this case, it's the seed set of the actual number of viable seeds, because all those non-viable seeds don't really matter from the standpoint of reproduction. And once again, not surprising, the other one wasn't significant. So why on earth would this one be significant? And once again, you see that is not relationship. There's just way too much variation around the line for this slope to be significantly different from 0. So the inflorescence isn't influencing the total amount of seeds, but maybe the energy stores that are stored in the stem and whatnot are influencing it. And actually, as it turns out, the bigger the plant is, the more seeds they produce. And so it isn't the inflorescence size that matters, it's actually the size of the plant itself. And so once again, it's a significant result, and it's actually describing quite a lot of the variation. About 56% of the variation in total number of seeds is accounted for by the mass of the plant. And once again, we see that this is the relationship, and once again, it's just based on, once again, the smaller set of data that are mainly the Tuesday lab data. But you can, once again, just look at the viable seeds and see if that relationship still holds. But when you look at the viable seeds, then that relationship goes away. So it's producing more seeds, but it's not producing more viable seeds as the plant gets bigger. And that actually looks very similar to that. Because these are all around 70 to 90% in terms of percent viable, so you shouldn't expect these two figures to differ that much. One of the other things that you might expect to see in any organism is this size number trade-off. Does the number of seeds that you produce, is that negatively correlated with the size of those seeds? Because you can either produce a few large seeds or a lot of small seeds, and this was disappointingly not significant as well. I don't know why there's a summary data in there. And this plot looks as flat as a pancake, if I recall correctly, yeah. So if you produce a ton of seeds, you aren't really sacrificing seed size at all, probably because these seeds are so tiny to begin with. So larger plants produce more seeds. Where they get this energy? Do they get this energy from leaves? So is the leaf area related to the dry mass? These things are not correlated, even though you might expect them to be, because the total dry mass is partly made up of leaves. And so, but the leaf area isn't contributing to the mass of the plant. And once again, a virtually flat line. But perhaps they're just using their seed, the energy, not for growth of the plant, but for seed production. And once again, this doesn't have a relationship. And so whatever the leaves are doing, it's not being allocated differently between growth and reproduction. Seems to be putting energy into both of them. So if we just look at viable seeds, once again, the story isn't any different. So maybe the way in which plants succeed reproductively is just by producing a ton of seeds. And as a result of putting out a ton of flowers and putting out a ton of seeds, when a certain proportion of those seeds are non-viable, the way to just get more viable seeds is just to produce more seeds in total. So this is going to look at the total number of seeds that you estimated for the entire inflorescence versus the number of viable seeds of that total seed set. And this shouldn't surprise you, is going to be totally related. A very highly significant p-value, and the R-square value is really high. And the reason for this is that plants that produce more seeds also produce more viable seeds, simply because every plant is producing 75 to 90% viable seeds. And so you would expect this to be, a correlation, but it's because the percent viable is relatively constant across the range of total seed production. So you would expect this next correlation to not be significant at all. And it turns out it's not significant at all, and it's really, really not significant, a really high p-value and a really low r-square value, because when you plot it, you see that every plant, regardless of the total number of seeds produced, is producing between 65 and 90% of the seeds they produce are viable, which means that 10 to 35% of them are non-viable, which then goes back to thinking about things like pollen limitation. Is this a species that tends to be limited by the availability of pollen? This type of data indicates that perhaps it does. One of the things that you might think about as you talk about this, both in the pollen limitation group and in the life history group, is a good follow-up experiment would be to do a study where you actually test pollen limitation more rigidly. The pollen limitation group, we've talked about this quite a lot. How would you do that? Anybody from the pollen limitation group? Where is the pollen limitation group?
I'm here.
Can you repeat the question?
Yeah, so it appears that there's maybe a lot of pollen limitation, because in this case, 35% of the seeds that are produced are not viable, probably because they didn't get pollinated. So you guys were trying to look at pollen limitation by looking at pollen on stigmas. The life history group isn't really looking at pollen limitation at all, but have some data that indicate that maybe these plants are pollen limited. What's a stronger way of testing pollen limitation than looking at pollen on stigmas, which is what you guys did? I'm still with you.
Is this when we were talking about putting the nets or the bags over the individual flowers?
This is exactly what we were talking about when we were talking about putting nets and bags over flowers. When you put a net over a flower, what does that prevent?
It prevents other pollinators from pollinating the flower.
It prevents other pollinators from pollinating. So if you get any seeds from those flowers that are netted, that's all due to what?
Self-pollination.
That's all due to self-pollination. We expect these guys to be self-incompatible, and so we would expect relatively low seed set. But that gives us a baseline. If we put a net around the inflorescence and we get X number of seeds out of that, then that's baseline selfing rate. Then we would look at another set of inflorescences that are not netted, and we would look at the seed set from that group, and we would say, okay, that's the seed set that you would get when you have access to pollinators. A certain proportion of that seed set, though, is also due to selfing, and so then we would be able to calculate, well, what proportion of the total viable seeds are produced due to self-pollination versus due to pollination from other plants. And then we would be able to say, okay, pollination from other plants is coming in at this rate, producing this number of seeds. And then we would then be able to say, okay, well, then the non-viable seeds, or whatever could have been pollinated, had there either been more selfing or more insect pollination. And so you would get a firmer idea of what pollen limitation is by basically saying, what's the production when you have insects available? What's production with insects? And then of that production, how much then doesn't get converted into actual viable seeds? So I would say that there's, a good case that these plants are at least somewhat pollen limited, both from the life history data and also from the stigma data, where you're just not seeing very much pollen on the stigmas at all. Oh, and that's the end of that analysis. questions from the life history group? Your basic idea is that if you have more energy available to you, you can build bigger plants, or you can build bigger flowers, bigger inflorescences with more individual flowers, and those things might actually contribute to the number of seeds that you produce. But it turns out that a lot of those relationships just didn't pan out. Being a bigger plant gave you bigger inflorescences, but those bigger inflorescences didn't necessarily translate into higher numbers of seeds and things like this. So for you guys, I think what would be useful would be to create a flow chart that basically shows you kind of how you expect, the main drivers of what it is to be a plant, the size of the plant, the amount of leaves the plant has for doing photosynthesis, the size of the flowers and things like that, how those interact with one another, and also then might influence seed set. And then maybe on that figure, put which of those relationships were actually significant in ways that you would expect from life history theory to be important. Bigger plants produces bigger flowers. Okay, that's important perhaps. But then if a bigger flower doesn't produce more seeds, then all that investment in being a bigger plant and being a bigger flower doesn't really gain you much in terms of reproductive success. Another productive way of thinking through how you want to talk about this would be to print off all of the figures and lay them out and rearrange them so that you kind of walk yourself through the progression of how is plant size related? Plant mass versus plant height, are those related? Sure they are, that's just general size issues. Well then how does the size of the plant then influence the size of the inflorescence? If those relationships are significant, then does that then have an effect on the number of seeds and the total seed set, things like that? And having the figures laid out in front of you might also help you with that process. Questions?
For the figures that we didn't have data from Thursday's group, do we need to acknowledge in our presentation that results could have been different?
Yeah, sure. So for all of these things, let's go back and look. For all of these things, having more data might have ultimately mattered, because you'll also see that whenever you have a low sample size, you have low power to detect the slope of the line. And so not surprising that for some of the things that you found that were significant are also the plots that you have the most data for. So total dry mass for inflorescence mass, there are good biological reasons for expecting those two things to be correlated, but it also doesn't hurt that you have a much fuller data set for that relationship. Same thing here, total dry mass on inflorescence length, good reason to expect that would be correlated, but once again, you detect it because, at least in part, that you have larger sample sizes. Same thing here. When you get to some of these other kind of finer grain nitty-gritty things, like if you're a bigger plant and you produce bigger flowers, you might expect to see more seeds. Well, you see this thing trending upwards, but there's all this variation around it such that you could fit a line through the middle of this that is a horizontal line. And so having more data might have led to a different result simply because it would have given you more power if the relationship was similar in the data that you don't have, but you don't have those data. So yeah, sure, that's a valid point. As we approach doing this perhaps next year, one of the things that we're going to be trying to do, Dr. Judd and I, is just keeping a closer eye on what the different groups are doing to make sure that all of the data from all of the groups are being collected and they're all being collected in the same way. So it's useful sometimes to go and look at the actual data file. this was life history full. So let me pull this up onto the screen. So here's the Tuesday lab. You did fewer plants, but the plants that you did more completely. The Thursday lab did more plants, but a lot of the really important things for life history, they're just missing. So you don't have seed counts. And the whole point of looking at life history theory is to see how these various aspects of the plants impacts seed production, reproductive output. And so when we get to these later comparisons, we're basically just dealing with this portion of the data set, because the Thursday lab is absent for whatever reason. I'm not sure what happens here. So yeah, that's a totally valid point. So I would suggest that you also go and look at the data files themselves in addition to looking at the analysis, because it will give you some insights into, oh, well, why did these things turn out this way? Why are these figures populated with relatively few data points while these other figures are populated with a lot more data points? Well, it's because of the completeness of the data set. And more data gives you more power for detecting things like whether or not your slope is different from a slope of 0. Does that help?
Yeah, thank you.
Sure. Other questions from the life history group?
So I know that we talked a lot about like the size number trade-off in like our little presentation that we did. Do we
You
still
did.
need to like address that fully, even though like we didn't find a like correlation?
Yeah, and I was talking to Dr. Judd about this last night as we were talking about your data. Dr. Judd and I were working on this into the night. And he actually sent me something that I didn't have time to look at. This MNAT paper shows that a neutral or positive relationship between seed size and number is not uncommon, probably because research data swamps the signal. Okay, so one of the discussions that Dr. Judd and I were having last night is that maybe plants don't experience size number trade-offs, except in those plants that produce these big, gigantic fruits. So goldenrod is producing little tiny seeds. You guys were impressed at how tiny the seeds were. And so if you're reducing the size of the seed down to that point, chances are you have so much surplus energy from photosynthesis that you could produce as many of those things as you wanted to without having to make them any smaller than they currently are. And so he found a paper. I'll just send this out to you guys. That basically says that there isn't a size number trade-off in many plants because of this reason. And so we would expect to see size number trade-offs in things like acorns and walnuts and oranges and apples and things like this, because there's really a lot of investment of the total energy of the plant into reproduction. But because these seeds are so tiny, you might expect that the total energy stores that the plant has available to it are just huge relative to the amount of energy that they're putting into reproduction. So you wouldn't expect to see a size number trade-off. Does that make sense?
Yeah, so should we talk about that like in our report as well?
Oh yeah, yeah, totally. So when you're writing the caption for that particular figure, put an explanation in there as to why you think that result isn't a sample size issue. That result is actually probably just the result.
Okay, thank you.
And I'm going to send you this email from Dr. Judd. Yeah, so you're also going to see some of our conversations trying to rectify your guys' data, but that's fine. Okay. So a couple of papers in there. One of them has to do with size number trade-off, and one of them has to do with storage and rhizomes. So remember that this is a rhizomatous species. It reproduces actually by sending out rhizomes, but those rhizomes are basically underground stems, and these underground stems are where you can store a lot of energy. Does it shock you that we think about your projects on an hourly basis? Other questions for Life History Group? All right. Then the last group is the gall group. So for those of you not aware, there are these flower galls that occur on the goldenrods, and we had one group that looked at life history characteristics of the galling plants. And we did two things with the gall data set. One thing that we tried to do was to get enough data out of the two groups to actually do something with it. And then the other thing that we wanted to do is we wanted to compare the life history of the gall group with the life history data from the life history group, which presumably wasn't looking at flowers that had galls on them. So in this group's script, there are going to be some things that are somewhat different. So load up your packages, and then we're going to read in the whole gall data set. And the gall data set is problematic in some ways. Let me go and get it. Oh, that's the wrong place in my life that I want to be. So the gall data were difficult for a variety of reasons. But so for example, in the Thursday lab, they got a lot of the seed data. But in the Tuesday lab, they were lacking a bunch of the data on viable and non-viable seeds. And then what we did was we appended the life history data to the bottom of this. So we've combined two data sets that hopefully we're collecting things in similar ways. Although in terms of inflorescence dry mass, one group reported it as milligrams and the other one reported it as grams. And so I had to go back and calculate things weirdly when I got a graph that didn't make any sense to me. So the fact that there's a certain amount of missing data, both in the Thursday lab and in the Tuesday lab, once again, gave us some sample size issues. But you know, you get the data that you have, and then you kind of work with those data. So once we have this, I did some of the same calculations where we calculated seed size, the percent viable, and the percent and the seed set in the same way that we did it for the life history data set. And so you can now see that those things are added: seed size, percent viable, and seed set are all in there. That's weird. And this is the last script that I was working on, so it's actually the roughest. Although it's pretty good until the end. And then the end things go drastically wrong. I need to go pull something up and look at something because something doesn't seem right to me. Okay, all right. I'm gonna do some corrections on the fly. The original data set had a column for seed set, but in some cases, we were able to calculate seed set from the percent viable and non-viable to generate this. And it looks like the values generated in R are different than the ones that are in the original Excel file. And then because we didn't have percent viable and non-viable for the Tuesday lab, all we have is the total count of seeds, which probably overestimates seed set because those seeds are going to have some non-viable seeds in there. So that's one of the caveats that you guys need to make, is that when we get a seed set value here, let me see if I can do this way. Yeah. So when we get the value here, so this is the R output. So you see it's 4054. Now this is not behaving. So you see 4054, 4326, 4339, 7167, and then you get down to 1629, 6049, 5176, 3693. So those are the seed set data, but if we do seed set 2, we're going to see that probably some of these things don't match up, which is 4054.6, 43.26, 339, 7167. How can those be different? Oh, that is not right. Ah, that's the reason. Okay, all right. That's why it messed up. Okay. Give me a minute. I'm gonna have to do something. Let's get on this line. I hate Excel. Hating Excel is an okay thing to do, I think. All right. that's what happened. That is what happened. All right, I have to go back in. I'm going to not calculate seed set in R. I'm going to use the Excel file to do this. So I have to reread this in. So now seed set is there and has 21 values. But it should have way more values than that. Why are those not registering as numbers? This is the difficulty. So many things. That's going to fix the thing at the end, I think. Ah, they're all in now. Okay, cool. Awesome. All right, fine. Sorry about that. You guys still with me?
Yes.
Okay, good. Yeah, sorry. It wasn't recognizing some of the data. So, the gall data are going to proceed in kind of two phases. One phase is just going to look at the gall data by themselves. And so what we want to do is we want to take that combined data set and divide it into just the gall data, and then we'll look at the combo data set at the end. And so this, if you do a summary of the data, you see that in the column galled, there's 23 galled and 38 non-galled. I'm sorry, 23 non-galled and 38 galled. Well, we want to get rid of the ones that are not galled. And so the exclamation point basically means to exclude. And so we're gonna create a new data set called DAT1 that subsets the original data file, but it gets rid of all of the non-galled, individuals. And so when we go back and summarize DAT1, we see that galled, there's 0 non-galled and 38 galled. And so that's the data set that we're going to work at. So this is going to look very similar to the life history group analysis, because many of the same kinds of questions that they're asking are similar, like how does the size of the plant influence seed set? But they're then going to want to know, well, then how does galling reduce that seed set to a certain degree? So we might look at how plant height influences inflorescence length. And we find that those two things are not correlated, although they were correlated in the life history data set. And once again, if you want to convince yourself of this, it's a virtually flat line, but one of the things you might notice is that there are two short plants that have really long inflorescences, and Dr. Judd has called attention to it right here. So now we can dig a little more into detail in terms of reproductive output. Does plant height influence the total number of seeds? And you'll notice that we're not using mass in any of this, and it's because the galling group didn't measure mass, and we should have asked them to measure mass. I don't know why we didn't ask them to measure mass, but we didn't. But it turns out taller plants produce more seeds. And once again, part of the reason that you can detect this is because you have a decent number of data to put to it. Do larger plants have higher seed set? Now, this is the seed set data that is based partly on multiplying percent viable times total seeds, but also is partly on just a total seed count. So this is a little sketchy. And what we see is that taller plants do produce higher seed set. And once again, we can see what that looks like by looking at that. And so taller plants were more viable seeds or a mixture of viable and non-viable seeds. So it makes sense that longer inflorescences would have higher seed set simply because they probably have more flowers. So we can look at inflorescence length versus seed set. And we find that there isn't a relationship between that. So the length of the inflorescence doesn't influence seed set at all. So it really must have something to do with something else other than the total number of flowers that are there. Because branching patterns vary, we might look at mass of the inflorescence rather than the length of the inflorescence. And so maybe the reason that this is not significant is that if you have a short inflorescence that has lots of branches, it may have as many flowers as a longer inflorescence with fewer branches. So dry mass gets rid of the effect of branching. And when we do this, we find that dry mass is a really strong predictor. Really strong predictor. The R-square value is 92% of the variation in seed set is accounted for by inflorescence dry mass, which that's huge. And so when we look at that, that's what a really nice regression looks like. So the bigger the inflorescence is, the more seeds you have. But now the question is the seed set affected by galling? And so for this, knowing the number of branches that are affected matters. And unfortunately, one of the classes didn't measure the number of branches that were affected. And so this is going to be, by definition, a smaller data set. And we find that even though it's a smaller data set, it's still significant, although the R-square value is not particularly high. And so, so this is actually not correct because this was done on a smaller data set. Okay, so these are some things that I'm clearing up from earlier analyses that didn't have both sets of data in it. So when we look at this, we see that as the ratio of golden fluorescence branches increases, that's weird. I didn't pay attention to the slope. The slope. Whoa, that's weird. The more galling you have, the more seeds you produce. That is an unexpected result. Okay, so this is a totally unexpected result. And like I said, this was the last one I was working on, and we didn't get aspects of the data rectified until late last night, which is why I'm discovering these things in real time. So the more galling you have, the more seeds you have, which is really not what we would have expected to see. Oh yeah, so he observed it. He observed it in his early analysis as well. So we might be seeing the effect of inflorescence size or mass here. We have to compare life history data. So this was an unexpected result. So maybe when we look at the life history data from the life history group, it might shed some light on this. So there's this thing called the target effect, which basically means that when a parasitoid is looking for a host, it might just go to hosts that are easier to detect. And so if inflorescence branches, if the size of the inflorescence makes it a bigger target, you might expect the number of branches affected to be correlated with that. So we can look at that, and it turns out there's no relationship there. So larger inflorescences do not have more gold branches, and this is what it looks like. And so is there a target effect at the patch scale? So if the number of branches are affected by the number of plants in a patch, that might also matter. And so that is not significant though. Once again, a high p-value and a really low R-square value. And that's what this looks like. So, and this is where I got screwed up early this morning. So the last thing we might want to do is we might want to look at both ungalled and galled plants. And so we're going to look at the combined data set now. And we want to answer the question, does galling affect the seed set? And the way you do that is you do that by comparing the reproductive output of galled plants with the reproductive output of ungalled plants, but you want to correct by something that is meaningful. And so the thing that you might want to correct by is something that had significance earlier, which was inflorescence dry mass. So this analysis does an analysis of variance where it looks at differences between galled and ungalled plants in terms of seed set, but it statistically removes the effect of the size of the inflorescence. And I got this to work earlier today, and then I made some changes to the data file, and then it didn't work. So we're going to see what it does. Oh, it did exactly what I expected it to do. So there is an effect of being galled or not, and of course there is an effect of the regression. So now I want to see if I can get this to give me a meaningful graphic. So here's what you see is if it's galled plants have generally fewer seeds than ungalled plants, which is exactly what you would expect. You would expect a parasite that infects the flowers and destroys the flowers essentially in order to create an area for the offspring insect to live. You would expect that all of those flowers that get taken out of production are going to reduce the overall reproductive output of the plant. And in reality, that's what we see. And that effect is especially true for smaller inflorescences than it is for larger inflorescences. And so it begins to make me wonder about if the mass of this inflorescence is correct. And so we keep seeing this outlier over and over and over again. And it makes me wonder if that's an actual value or if that's something that got miskeyed. In one of the other data sets, or maybe it was this, no, in one of the data sets, somebody had keyed in a 62 when it should have been a 26. And that's a common mistake to make when putting data in. But this is exactly what you would expect to see in plants that are affected by a parasite that destroys flowers would have lower seed output than plants that are not affected by that parasite. And this is even considering that this measure of seed set in the galled plants probably includes some flowers where we're not actually measuring the seed set in terms of the number of total viable seeds, but it's actually just total seed production. And so even with that caveat, these are lower than that. So this is an analysis of covariance. What it attempts to do is it attempts to use the relationship between inflorescence mass and seed set to correct these values to some grand average. So if you look at what the average inflorescence mass is across all of the data set, it's somewhere here in the middle. What analysis of covariance does basically is it moves each of these data points up to that grand average parallel to the line, and it moves these up this line parallel to its line and creates a cluster here at the grand mean and a cluster here at the grand mean. And this cluster is going to be higher than this cluster after bringing them. It basically says, what would the size of the seed set be if all of the plants had an average inflorescence mass? And so it removes the effect of inflorescence mass influencing seed set. It says if all flowers had the same inflorescence mass, what would the seed set be? And it would be down here for the galled plants, and it would be up here for the ungalled plants. And what I'll do is, since we haven't taught you about analysis of covariance, I'll write you guys up a little explanation as to what exactly the analysis of covariance is doing. But really, this is the most statistically appropriate way to look at these things. And this is cool because this is exactly what we expected to find. And why we had one group working on non-galled flowers and another group working on galled flowers. Questions about this data set? Gall people, talk to me. Are any of you there?
Yeah, I'm here.
Okay, good.
I think I get it, you know. I just want, you know, I just have to go back and analyze these graphs more.
Okay. Connor, are you in the bowling group?
Yeah, I am.
Okay, all right. I'm thinking of Cameron, sorry. Yeah, so I think the most unexpected result was this one, and Dr. Judd and I were talking about it last night, and because he mentioned it to me, and I don't have a good explanation for this, but even though the galling seems to increase seed set, it's not increasing it to a point that you can overcome the deficit in seed set that is experienced by galling when you compare galled plants to ungalled plants. So many of the same kinds of basic life history characteristics that you saw in the life history data set, like size of plant influencing seed set and things like that, those still hold for the galled plants, but the galled plants are just in general putting out way fewer seeds than ungalled plants. All right. Hopefully, while we were going through your group's data, you were paying careful attention. And when we were going through other groups, you were paying perhaps less intense attention, but you still got some salient points across. My advice is to, once I get this thing uploaded to Moodle, which will be momentarily because it's just a matter of uploading the files and clicking the submit button, download the R script, download the data file that corresponds to your group, and then run through the R script, and then contact me as you run through that when you're having problems, when you're not understanding something that you're seeing. And then we've produced a lot of graphics in all of these things. That doesn't mean that you have to talk about all of the graphics in your talk. It doesn't mean that you have to talk about all the graphs in your report when you choose graphs to put in there that you're going to append captions to. You want to figure out what's the story that we want to tell, and with that story that we're trying to tell, which graphics are the informative graphics to get that idea across. Which is why I think for the Gall group and the life history group, it's important to go back to basic principles and say, what are the things that a plant does that will then influence seed set? Is investing in the size of the plant going to influence seed set? If it does influence seed set, how do we expect that to do? Do bigger plants, are they able to produce bigger inflorescences? And as a result of that, there are more individual flowers, so that gives you bigger seed set. So working out those relationships to tell you which results that you want to pay the most attention to, that's going to be really useful for you, for both groups. And then the goal group has the additional thing here at the end. We have data from the life history group that we can apply to our goal data and ask the big question about the whole purpose of your group existing was, does this galling, this parasitism by these insects, does that negatively affect the reproductive output? And it clearly does. All right, it's been a rough couple of days. I know that your heads are about to explode. I guess I would probably get a lot of thumbs up about that at this point. Give me a little while to get these things uploaded, and then they'll be available to you. And you can start churning through them yourselves. And just get back to me or Dr. Judd with any questions that you have. I wrote 3 of the scripts. Dr. Judd wrote the goal script, but he used my life history script as a starting point. So we're both pretty familiar with the scripts, and he's unfamiliar with the last part of the script because, once again, we didn't have the combined data set of the gold and non-gold data until early this morning. So, but yeah, just let me know if you have any questions, or let Dr. Judd know if you have any questions as you go. And start getting together with your group to figure out what your story is going to be. What's the narrative line that carries your world presentation through? That narrative story that you have, that will then influence what you talk about in terms of methods and what you talk about in terms of results. Hopefully, the gecko presentation that I made for you earlier today had a good narrative line through it. It had a story to tell. The best presentations have a good story to tell. And then the graphs and everything else are just employed in the service of making sure that that story is clear and compelling to people who would be listening to it. All right. If that's all and there are no other questions, we are done.
Thank you.
I
Sure.
have a question for the research summary.
Oh yeah.
due to Friday. So you're not in your office, but I want to come to your office hour. Can you send me the Zoom link?
Yeah, I'm meeting with Connor at 11. Are you asking me about quantitative genetics, Connor?
That too, and the Broody paper due this Friday.
Okay. Do you want to just both come at 11? We'll hash it out together, the two
Yeah.
of you. Is that all right with you?
Yes. Yes.
Okay. I'll send you the
and-
Zoom link. Yep.
Okay, thank
Go
you.
ahead.
And also, can you tell me when will we have to submit this research paper?
Oh, that'll be due on Tuesday when you come to lab. But it's going to be the introduction and methods that you've already written, and you're just adding the results to it. So you're going to be doing the adding of the results to your thought process as you go through and present the presentation. You're not going to write a results section formally. You're going to just present the results in figures with captions attached to them. And there is going to be no discussion section.
Okay. And
Okay.
we don't have to write discussion.
Don't have to write a discussion. You'll tell us what your discussion is in the presentation in class.
Okay. And well, during the lab period, we like our research group texted each other, and we also talked to like a different group, and we all told each other that you forgot to tell us that the introduction and the method is due today. So we didn't even know about that.
Oh, I could have sworn that I covered that in the last time we met before the break.
I don't know, like both of our group didn't know that. So it was kind of awkward today that we didn't know we have that too.
Okay. let me see. So you were assigned, I'm trying to think how long ago it was. You were assigned to write an introduction and a method that you would then bring to class today with the intention of exchanging it with your peers. And I just don't remember what week we assigned that in lab. It was before Thanksgiving that we assigned it. And I can't remember if it was the week before Thanksgiving or the week before that we assigned it. But yeah, I know that I mentioned it in lab because we were talking about, that was when we were going to have you write a full report and we wanted you to get started on it. So if you didn't get that, it's just because it got missed in lab, because I know for a fact that I mentioned it. Let me think.